Alright, today's lesson, we're gonna focus heavily on CRISPR-Cas9, the gene editing tool that we've discussed in the past, and how it is applied in the context of agriculture, specifically in the context of photosynthesis and plants, right? So we've been looking at photosynthesis, all the factors that play into photosynthesis, and all the different prerequisites of photosynthesis. Today, we're going to look at what can we do to possibly make photosynthesis better or make other features about the plant better so that there's a agricultural application for our benefit. So essentially, what can we do using genetic engineering to make agriculture better for the sake of humans? That's what we do today. All right, first things first, what are the factors that could, oh yeah, and I totally forgot to do attendance again. I have to restart it. All right. Abby, Gavin, Yasmin, Nair, Yakut, Janita, Cyril is late. It's going to be very late, apparently. Iman, and Melinda, and Zoe, Tan. All right. Starting now, right? So, just start off like this. So today's lesson is going to be about agriculture. Yeah. All right, moving on. So, what are the factors that could increase or decrease photosynthesis? So, this is basically the topic of the previous lesson. Can I get Yasmin? Give me one. Light intensity. So, that one could be light availability, presence of light, anything when it comes to do with light. That's one of the factors. Okay. Second factor, Gavin. Oh, you weren't here. Emelinda? Temperature. Okay, because temperature has two effects. What are the effects? Can you tell me? So it plays on two roles, temperature. What does temperature have to do with normal enzyme activity? What would it do if it's too high? What's the word? Denatured. Okay. If it goes too cold, it'll freeze, right? That's one thing that temperature has an influence on. Another thing has to do with one of the specific enzymes. Who can tell me? Okay, there's an enzyme that the temperature will change the properties of. So,
please excuse the interruption. Student Lorena, please go immediately to the bus, which is waiting to depart. Lorena, to the bus, please.
So, temperature also changes the properties of a specific enzyme to make it have a higher affinity towards another molecule. So, which enzyme am I talking about? And what molecule is that? Can I get Yasmin? So higher temperature changes enzyme that is part of photosynthesis to make it have a higher affinity towards oxygen. Yeah, Rubisco, right? So that's two things that temperature does, and you need to be able to link it back to those two things. And can I get another factor that affects photosynthesis, Janita? pH level. And Abi, another one? Substrate concentration. So rather than saying substrate concentration, you should say CO2 levels, right? Because you need to be specific about this, especially when you're referring to photosynthesis, right? And there's 6 more. How many have I heard so far? 3, 4, 4. Okay, Ayman, were you there? No, right? Yeah, put water. And lastly, 10. What was the last factor? No? Zoe? Okay, what role does oxygen have in the activity of Rubisco? What would we call that oxygen as? If CO2 is a substrate, what is oxygen in that role? There's a specific word I'm looking for. this has to do with enzymes, right? What binds to enzymes and inhibitor? So enzyme inhibitor, that's the last factor, right? There are 6 factors. Each of them affects photosynthesis in a different way, and you need to be able to mention them. Why am I asking that question? Sorry, answer. All right, this question. This is an example of a question that's going to get you guys to look at it very specifically. So when you're answering this question, my biggest recommendation for you guys is, rather than looking for which one is the correct one, go for the ones that are wrong. Identify mistakes first, because it's easier to find mistakes than to prove something correct, right? For example, is there any mistakes that you can spot in A? Yeah, sorry? NADH. Yeah, so NADH is not part of photosynthesis. NADH is actually part of cellular respiration that humans do. So humans that mitochondria does. So this is immediately wrong because it is not describing photosynthesis. Like I said, right? One mistake is all it takes, then you can make the entire answer wrong, right? Next, let's look at B. Who can tell me if there's a mistake here? Someone else. Come on. Abi, what do you think? You know what? I'm going to give everyone 2 minutes to try to answer this question. And I'll see whether everyone can get the right answer. Make sure you look at the inputs and the outputs.
Sorry for interruption. Good year 9 student, Lorena Bruto, if you're still in college, please come to the office now. Lorena, please come to the office.
All right, let's get a poll, shall we? Who thinks the answer is D? Raise your hands. Who thinks the answer is C? Raise your hands. Who thinks the answer is B? Raise your hands. All right. So if I ask you what's wrong with B, who can tell me? If like no one answering B, then there must be something wrong with it, right? What is it? Yeah, sorry. So let's look at the direction of the arrows, right? So ADP is becoming into ATP, NADP plus is becoming NADPH. So if this is the light dependent reaction, that's actually showing the right transition, right? It's becoming charged from the light dependent reaction. They intentionally made it look like this just to confuse you guys. The actual answer, the answer is B. Okay, so let's identify what was wrong with the other answers. And like I said, this kind of questions require you to pay a lot of close attention to the details, right? Here, the detail is right here. This one point. So ATP is becoming ADP from the light dependent reaction. Immediately wrong. That's where they all get charged. This is a transition to become uncharged. So it is incorrect. For this one, it's at the products. See what is released from the light independent reaction. This is oxygen. And where is oxygen actually released? Can I get Janita? Sorry, 10. Where is oxygen released in photosynthesis? The first stage or second stage? Where is water split up? First stage or second stage? So water splitting up is what releases the oxygen, and that's why it's generated. So it's all in the first stage for oxygen, okay? So always remember that, and always make sure you pay close attention to these kind of details. Essentially, these kind of questions require you to pay a lot of close attention while also making sure you're not wasting too much time. So they're intentionally designed to trump you. So when it comes to these kind of questions, it is always going to be harder for you to find which one's the correct one, because that takes more time to verify every step. Rather, go through each quickly to see whether there are any incorrect details. It's quicker to find things that are wrong rather than proving the entire thing is correct. So in each question, look at the key details, see if there's any mistakes, right? Look at the products, check all of them, see are they all correct. Then you can move on to the next step. Look at all the coenzymes. Are they becoming charged from the light-dependent reaction, and then used up during the light-independent reaction? When you do verification steps like that, quickly, you get to eliminate answers much more faster. Good thing we haven't started the lesson. I mean, we're running the review question. All right. Alright. Okay, moving on. So today we're going to look at how CRISPR, the gene editing technique, the tool that we've learned, is used in agriculture. And we've actually looked at a few examples of that when we learned about CRISPR. But today we're going to look more in depth into it and look at the different ways CRISPR can be used to modify a plant for it to become more efficient and more resistant and actually be better in terms of generating crops. So before we get into how it is done or how it is used, first let's revise how CRISPR-Cas9 works. So CRISPR-Cas9 is essentially a defense mechanism that evolved in bacteria to fight against bacteriophage, which are viruses that attack bacteria. How the CRISPR-Cas9 works is using two different structures that can be found on one sequence, right? So if this is the gene sequence, I'm already running out of ink. So if this is the gene sequence, the first thing you're going to find is a Cas9 gene. And what does the Cas9 gene code for? What do genes code for? Kevin, what do genes mean? Yeah. No, but gene is DNA, right? What do they mean? Proteins, right? So a Cas9 gene will make a Cas9 protein. Okay. So essentially that's an enzyme to begin with, but you can just generalize and say it's a protein, that's fine. And then upstream of that gene, you'll find a special sequence called CRISPR. This is a sequence that is made up of multiple spacers and repeaters. Repeat. And spacer again. And repeat. So due to the fact that they keep repeating over and over, that's why they're called clustered clustered regularly interspaced short palindromic repeats. CRISPR, right? Where do we get this spacer from? This is the key part that is used in the defense mechanism. These spacers are basically sequences from previous bacteriophage that have invaded the bacteria. So when the bacteria is able to survive that attack from another bacteriophage, it takes a segment of the DNA from the bacteriophage and saves it into the spacer. Basically, think of it like a reference where it remembers the viruses, the bacteriophage that has attacked it, and it keeps a copy of its DNA. So this is a form of adaptive immunity, which is similar to how your vaccines work, right? Basically, you keep a copy of the antibody that defends against that specific pathogen, and your body uses that to make more defenses later on. The same thing happens with bacteria. It saves a sequence from the invading bacteriophage to be used to cut out any similar copies later on. So how does that work? First, when a bacteriophage invades the bacteria again, from the spacer, a guide RNA is made. That guide RNA is essential in the next stage. Let me see. So, yeah. Essentially, first things first, bacteriophage invades the bacteria, and once the bacteria survives, it saves a copy of that bacteriophage DNA into the spacer part of the CRISPR sequence. And when another bacteriophage invades the bacteria will make a guide RNA, this molecule, using the CRISPR sequence, specifically the spacer sequence, where it makes a RNA copy of that spacer sequence. Okay, using that G guide RNA and also the Cas9 protein that comes out of this, they both will bind to each other, creating a Cas9 complex. Sorry, CRISPR-Cas9 complex. And this is the tool that will actually go through each cell, go through each part of the cell looking for complementary DNA. When the Cas9 protein and the guide RNA join together, they form the CRISPR Cas9 complex. And this complex is what is responsible for actually searching every part of the bacteria for a complementary sequence to the spacer. Because remember that the spacer is a sequence that came from the original bacteriophage. So why does it do this? Why does it make a guide RNA, join it together with Cas9? Is that it's looking for a bacteriophage that has the same sequence. And if it finds the bacteriophage with the same sequence, it will make a cut there. And how it does that? Essentially, right? First thing first, they both bind together, they form the complex, then they go looking for all the sequences that can be found in the bacteria. It searches through them one at a time to make sure that there are no complementary sequences that it can find. If it finds one, there's two things it needs to do before it can be sure that that's a complementary sequence. The first thing is that it identifies a the first recognition site, which is the PAM sequence. The CRISPR-Cas9 complex identifies the PAM sequence in the DNA of the bacteriophage, and then only the second verification is done, where it checks for a complementary sequence against the rest of its guide RNA. So first thing, PAM sequence is checked. Second thing is checked against the sequence on the guide RNA to see whether it's complementary with the sequence that is searching through. If the both are complementary and both have passed the verification, there will be a single double strand cut through the sequence about a few base pairs in front of the PAM sequence. This will cut through both double strands because there are two active sites on each side of the CRISPR-Cas9 complex that can cut at both sides. And what kind of cut can we expect from this, Ayman? So there's two kind of cuts that can be made, sticky and blunt end. What kind of cut does CRISPR-Cas9 make? Sorry? Blunt cut? Are you sure? Yeah. It's a blunt cut, right? Essentially, there is no overhanging for the for the strand. There will be a direct blunt cut made. So how do we use this for genetic engineering? Is by basically modifying two major things. One, we modify the guide RNA ourselves. In other words, we make it ourselves because that is what the protein, the enzyme is using to search through the sequence to find a complementary sequence, right? So by modifying that, making it ourselves, we can basically program the protein to go through a sequence and find the specific spot we want it to make a cut, right? Because we already know the sequence to expect at that spot, so we can make a sgRNA, a single guide RNA that has a complementary sequence to whatever part of the sequence we want to make a cut at. Okay, because we already know where we want to make a cut, so we make a single guide RNA to go to that specific spot, find it complementary, and then make a cut there, right? So when a cut is made, what we do with it depends on what our intentions are. If we want to delete a gene, we can delete a gene. If we want to insert a gene, we can do that as well. But essentially, by modifying that and another thing, which is making sure that the Cas9 enzyme has a specific target pan sequence. Because there are two things that a CRISPR-Cas9 verifies before it makes a cut, right? First thing it verifies is the pan sequence. And then the second thing it verifies is whether the single guide RNA is complementary. So both of those need to be modified to use the complex as a genetic engineering tool. So the first thing they modify is making sure that the entire enzyme has the pan sequence that is complementary to wherever you want to make the cut, right? So yeah. So a Cas9 enzyme with a specific target pan sequence is obtained. Then the second thing they have to do is make a guide RNA, a single guide RNA that is complementary to where we want to make the cut. And both of those will then make the enzyme look for the specific target and find it complementary through the PAM sequence and the guide RNA and make a double strand cut. Once that cut has been made, a gene is inserted and thus a transgenic organism, genetically modified organism is made. Any questions about this? Okay, if you do have questions, please let me know. Like I said in the past, CRISPR is one of those things that you should really try to understand. This essentially, once you understand the overall system, we can simplify it to very simple steps, right? First thing, this first step is important to note that it happens at a different stage. Basically, think of it as you getting chicken pox for the first time in your life, right? This happens at a separate time, and then you get the immunity, and then anytime the strain tries to invade you, you are protected against it. So this needs to happen first, and it needs to be saved into the CRISPR sequence, and then only next time when there is a invasion of a bacteria, invasion of a bacteriophage, it can protect itself. Moving on. Okay, everyone understands CRISPR, I assume now. All right. So today's lesson, like I said, we're going to look at the potential uses and application of the gene editing technology of CRISPR-Cas9 to improve photosynthetic efficiencies and crop yields. So how will CRISPR-Cas9 be used to influence the crop's genome to change things that we want to change? So essentially, by changing the genome of agricultural crops, we can get a lot of function out of it, be it improving photosynthetic efficiency or even increasing crop yields in a variety of ways. We can even add nutritional value to the crops, meaning that we can make the crops have a higher nutritional value, so have more carbs or have more vitamins or have more minerals. So these are things we can program the genome to do, and all we need to do is specifically make a CRISPR-Cas9 sequence that can make the cut at exactly where we want to, and then insert the right gene or silence or remove the right gene to make sure that that happens. So, like I said in the past, genetic modification isn't done for kicks. Usually it's done for very specific purposes because of all the ethical, biological, and social implications. There are a lot of hurdles to cross to actually undertake genetic engineering research. So why is there such a strong reason for us to try to do this? Yeah, ma'am.
Someone can do that. And then we can go ahead and can.
So unfortunately, the thing about us is that every one of your cell has a copy of your DNA, like the entire copy of your DNA. So even if you modify a few of your cells, that region will become mutated, sure, but it won't spread like a virus. If you really want to actually modify an entire organism, who can tell me when the perfect time for that is? There's a specific point in the lives of everyone that you can modify whatever you want about them, and no matter what happens after that, they're going to be modified. Abi? So when you have a freshly fertilized egg, it's basically how many cells? 1, 2, at most it's 4 or 8 cells. If you modify those 4 or 8 cells to whatever you want, every cell that comes after that is going to have that modification, and you don't have to do anything about it. And essentially, after you do that, there is no going back, because whether the fetus, whether the embryo wants it or not, it's stuck with those genes. You can't undo it. In the same way, you can't modify your genes now, right? And it's like I said, like there's social, ethical, biological concerns. One of the social implications is that people start to use this technology so what's the word? So nonchalantly that it becomes about physical traits, hair color, eye color, skin color, muscle density, intelligence, these kind of things. Like you get to control what you give birth to, or what kind of offspring you have. And that's the kind of future that people are very, very afraid of, where things become very hazy between life and science. So there's a lot of reasons to be concerned about it, but at the same time, there's a lot of exciting possibilities as well. For example, CRISPR-Cas9 is a revolutionary tool to create gene therapy techniques. So something to help deal with genetic diseases, right? So something like cystic fibrosis, if you guys know, is caused by a genetic mutation where, if I remember correctly, it's one gene that causes one specific mutation at a gene causes cystic fibrosis. And through CRISPR-Cas9, they've actually released a therapy for it, not till this year, called CRISPR that actually treats for cystic fibrosis and thalassemia. And that's a very big deal because it's not something that was able to be done about 5 to 10 years ago. Okay, we have the technology to create therapies that can better human lives, but at the same time, it's such a powerful tool that you can be used to, further human chaos, if you will, right? So that's just me talking about people using it for vanity stuff, right? You haven't even considered how evil governments could possibly use this technology to create, excuse my term, but super soldiers, right? It is possible that science is there, increasing intelligence, increasing muscle density. It's all there. Whether governments actually would fund this kind of research, who knows, right? There's a lot of gray areas that these kind of sciences can exist in. Anyway, we're getting off topic. Essentially, CRISPR-Cas9 is a very powerful tool. How we use it is going to be very important because there's a lot of factors to consider and a lot of things that can cause it to become very touchy of a subject. Okay. So, like I said, there are reasons why people can actually undertake this kind of research because there are factors that are making it very important that we do this kind of research and be able to create crops that can have a better photosynthetic efficiency. The first reason is because C3 plants make up 85% of all plants on Earth, right? When it's 85%, it's going to cover a lot of crops as well, a lot of very useful plants and a lot of fruit-bearing plants. A lot of them fall under C3 plants. And the problem with that is that C3 plants have no adaptations against photorespiration, okay? And that means that if they are present in a hot and dry environment, they will not be able to undertake photosynthesis and will be carrying out photorespiration continuously. And if that continues on, the plants won't be able to make food and they will eventually die, right? Remember that photorespiration and photosynthesis are inversely connected. One can't happen without one can't happen if the other one's happening, right? If photorespiration is happening too much, photosynthesis can't happen. At the same time, if photosynthesis is maintained, photorespiration is not a worry. With C3 plants, if they do too much photorespiration, they can't do photosynthesis at all. That means they're not making any food. That means they will die eventually. And this is becoming more of an issue with climate change because due to the climate's changing in a unpredictable fashion, there are certain regions that are suddenly becoming too cold or too hot. Those regions were never prepared to deal with that kind of a transition, and the plants, the C3 plants in that region, will be the ones that are suffering. They have no defense against it because they have no adaptations for it as well. They can't do anything about it because as soon as it becomes hot and dry, photorespiration will happen. If it happens long enough, the plants will all die. Okay, even in this environment that we are in right now, if it gets too hot and dry, imagine not as bad as a desert, but something close, the plants around us will die very quickly because they can't do photosynthesis and they are undertaking too much photorespiration, and that causes them to die. So one of the reasons we have to find a way to adapt C3 plants so that they can actually survive in a hot and dry environment. Second reason, too many mouths to feed. So essentially, the human population is predicted to rise to about 11 billion and then only start to balance out or decline. So essentially, there are still more mouths to feed than ever, and that's only going to increase. But we are running out of arable land. Okay, this is a non-renewable resource, arable land, and we keep using it up, and it's only gonna increase because we are only having more and more mouths to feed. So the problem is, how are we going to be able to support that population? We need to be able to produce the necessary food to support that population, and we need to find a way to do it without the next factor is that we need to find a way to generate more food while also reducing the usage of land for crops, because we keep cutting down trees and clearing land to make food, and that is actually adding on to climate change, which is worsening the first factor, which is then creating a endless loop of problems, right? So we need to cut this cycle down somewhere, and one of the ways we can do that is by basically creating crops that can generate a much higher yield than it could before. Basically modifying the genes of the plant to make it make more food than it could before, so that we can basically use the same amount of land that we've been using to generate more crop than ever. Okay, that way we don't have to keep burning down forests or cutting down trees to clear land just for the sake of farming, because so much land has been used for that right now. So much land in Indonesia, Malaysia has been cleared out just for the sake of farming, and that has an effect, and that is going to just play into the system of climate change. Moving on. So essentially, we have the reasons of why we should modify our genes. So now we look at how we're going to do it. Okay. Essentially, if you simplified it, this is what it would look like, right? We have a regular tomato plant. We understand everything about its genome very well. Then we can think about how we're going to edit it. Once we know what we're doing, we know how to edit it. Once the edit has been made, for example, if you see here, literally one base was changed to from a T to an A, and that might have influence over the increased agricultural yield, meaning the size of the amount of tomatoes that are generated, how much photosynthesis it can carry out, and also how well it can grow. So essentially modifying it to have all these benefits for our benefit. Okay, that can be done with wheat plant as well. By understanding its genome, we can modify it such that it has increased efficiency, increased yield, and also increase its growth. So when we think about it, there are so many things we can modify in a plant to make it more efficient or more resistant, or just overall better for humanity, right? So there are a lot of things we can do. So let's look at each of them. So the first one is modifying the Calvin cycle. So just look at this one. So when I say modifying the Calvin cycle, this is referring to the second stage of photosynthesis, the Latin. We can actually modify the genome of plants such that we can modify rubiscose activity. So essentially, Modify resistance activity, we might even be able to prevent photorespiration, right? Another way to prevent photorespiration by changing C3 plants altogether into C4 and CAM plants. That is also possible with CRISPR-Cas9. Another thing we could do, introduce alternative routes to photorespiration. So instead of the plant immediately going into photorespiration, maybe we can introduce another pathway that is not as wasteful as photorespiration. So each of these are potential ideas, potential things that are being researched to be done to plants with CRISPR-Cas9 as a way to make them more efficient and better capable of surviving during climate change. So as you can see there, next is production enhancement, right? So this is talking about how we can make the crops better by modifying them, and that can be done through longer shelf life. Essentially, this is talking about something like the flavor saver tomato that I told you guys about when we were learning about CRISPR. That is the first genetically modified organism to be sold, and that had a gene that was silenced to make it last longer, so it wouldn't rot as quick as it used to. And that was done using a different technology. Essentially, we can do the same thing using CRISPR as well, where we modify or silence a gene to make it rot, or make the crop rot not as soon. Next is improving the nutritional value. Like I said, we can modify the crop such that it has a higher content of whatever nutrient we want. So it could be carbs, it could be minerals, or it could be vitamins. It's not that it's not that impossible to modify them to such that they have those kind of higher nutritional values. Another one is adding bioactive compounds. So that could be any compounds that could be useful for growth. For example, remember the golden rice that I mentioned and how they basically modified rice such that it became golden because they modified it in a way that vitamin A was being deposited into the rice, where that is not going to happen normally. This is basically them modifying the crop such that it has a nutrient that it never had before for the benefit of society. That can also be done as part of production enhancement. Next is plant organelles. So basically modifying the organs within a cell, a plant cell, known as organelles. So what can we do? First one is increasing the chloroplast efficiency. What do you think increasing the chloroplast efficiency would do? This is a question, Sarah. Okay, what do you think increasing the chloroplast efficiency will do? Where would it directly have an influence? Yeah, because what is happening at chloroplast? Yeah, I mean it's a site of photosynthesis, right? Yeah, basically, yeah. What does doing this do is that overall photosynthetic capabilities are more efficient. Second thing, adjusting stomata to improve CO2 uptake. Another factor of photosynthesis. Another one is improving light capture in chloroplast. Another factor of photosynthesis. So by modifying the organelles, we can make them basically uptake the reactants better. So for CO2, it'll take it up better or take more of it up. With light capture, it will absorb more energy from the sun and store it in the chlorophyll better. And the other ones, they are more about making the crops more resistant or tolerant towards external factors. For example, greater herbicide resistance and greater pesticide resistance. These are going to make sure that the plant can be sprayed with pesticides or insecticides without having to worry about them dying, right? So an example of which is the BT crop that we mentioned when we were learning CRISPR for the first time. These crops have been modified such that they generate a specific crystal-like protein that is basically toxic to pests. But they are so small that we can basically break it down ourselves. So it's a toxic pesticide that the plants themselves make that can be toxic to the pests and can be harmless to us. Another thing you could do is basically increasing the resistance to different herbicides and pesticides. That is basically modifying them so that they are not going to absorb those dangerous chemicals. They are able to survive or even use those chemicals for their own benefit. Next is disease resistance. This is basically improving the immunity such that they are no longer, or they will have immunity against viruses and bacteria, which again, like I said, by modifying your genome, there's just too many things that you could do. The possibilities are very much beyond your imagination. This is just a small sample, and this is specific to photosynthesis as well. There are so many things you could do beyond photosynthesis when it comes to modifying the genome, but this is just specifically to photosynthesis. That's why even then there's so many. And lastly is the physical tolerances. How tolerant can the plant be towards drought, sudden cold, and heat stress? Why is it important for a plant to have adaptations against heat stress? Can I get Zoe? But like, why? So in terms of C3 plants, why would it be important for them to be resistant to heat shock? Photorespiration, right? We don't want them to do photorespiration. And one of the factors that can cause photorespiration is a higher temperature heat shock. Okay, we don't want that to happen. So essentially, each of these can be done, and each of these are being researched, right? For example, researchers in the United Kingdom have been targeting the function of rubisco in tobacco plants, such that they want to use CRISPR-Cas9 to better understand the many genes relating to rubisco's complex function. So using CRISPR-Cas9 is not just about adding genes or modifying genes. It's also used to study genes. Who can give me an idea of how you'd use a gene modifying tool to study a gene? Any ideas? How do you think we use a gene editing tool to study what a gene does? So let's say I found a gene as I was sequencing a genome. I don't know what that gene does. I can now use CRISPR-Cas9 to find out what that gene does. How did I do that? What did I do? Why would cutting it allow me to know what it does? Exactly, right? By deactivating the gene, we can understand what downstream effects it has and reverse, basically go backwards to think about what could the possible function be. That is how genes are studied today, up until today. Basically deactivating the gene, seeing what effect it has, and go backwards from that. In fact, a lot of genetics is actually still done like that, where we don't really have other tools to study how genes play together. So the only way we can do that is to deactivate a gene and see what it does. So that's what they're doing here as well. They are deactivating certain genes and seeing how it has a effect on rubisco's complex function, and based on that, they can come up with the basic idea of how they all work together. So research showed that editing certain genes resulted in lower rubisco content and subsequently lower photosynthetic rates. All right, moving on. Is, like I said, production enhancement. Nutrients can be bumped up in a tomato, and they can also have an increased shelf life, so last longer. So when I say bumping up a nutritional content of crops, this might sound like something new that's been done, but this has been done for hundreds, if not thousands of years by humanity without using any complex tools. Okay, we've just been using a very old-fashioned tool known as selective breeding. Okay, basically, if you had a crop of 100 corn plants, 100 maize plants, you would pick the maize plant that got you the best crop and plant the same thing 100 times over. And if you keep repeating this process, eventually you'll get a final product that is very, very modified compared to how it was before, because you haven't modified anything. You're just artificially selecting for the best crop. And when you keep doing that over many generations, you will end up with a crop that was very different to how you started out. In fact, this is apparent with a lot of the fruits, a lot of the vegetables that we have, right? For example, corn, how it used to look versus how it looks now is super different. I feel. Sorry. What it looks like, and yeah, basically what it looks like. You can kind of tell, oh, okay, this one grew better, this one has more on. Yeah, okay. Oh, you don't have to go too far. This is what corn used to look like. And this is what it looks like now. And this is only because selective breeding was done. So years and generations of selective breeding corn to make sure it gets better and better every generation eventually led to us having what we have now. And this is the same with tomatoes, with apples, with so many other things that we don't even realize because we've been doing it as common practice. Like farmers don't even think about it as being, oh, modifying genes or anything. They just think about generating more crops or better crops. Yeah, what happened? No, go. Yeah. So even though we can still do this, we can do it much quicker now with CRISPR. We only need one generation to increase nutritional content. Even if we didn't have CRISPR, we are still able to do this. We are able to increase nutritional content of any crop by selectively breeding them. That is a very old tech, right? Old-fashioned technology, old-fashioned knowledge. Okay. So that is about improving the yield, improving the production. Next thing is, like I said, improving plant organelles such that they basically do photosynthesis better, right? They have a better chloroplast that can carry it out better. They have a better stomata such that it takes up of CO2 better. And then lastly is improving the light capture of the chloroplast such that it absorbs more sunlight to be stored and used later in the light independent reaction. And like I said, herbicide and pesticide resistance. So you can see here because this plant is able to make a specific protein, right? Because this plant is able to make a specific protein, unlike the one on the left, it can basically be resistant to whatever pesticide or insecticide that they use, and it essentially allows them to survive, whereas the other one would die. Because that one protein is the difference between it being able to neutralize or protect itself from the toxins versus not being able to do anything about it. Right? And disease resistance, yeah, this should be pretty straightforward. Physical tolerance. So this is becoming, like I said, especially important with climate change. All three of these are becoming more and more unpredictable, right? We don't know where it's going to be getting colder. We don't know where it's going to get hotter. And we don't know where it's going to be a drought. Okay, so it's going to be more important than ever to modify genes of plants such that they can resist these kind of sudden changes and be tolerant towards them, or else we're going to have mass die-offs of crops from these kind of changes. So for us to be able to modify a genome of anything, let alone plants, the first thing that needs to be done is that we need to gain a in-depth knowledge on the genome itself of whatever living thing it is. In the context of genomes of plants, we need to understand the entire photosynthetic process of the target crop species. If we want to just modify something related to photosynthesis, then we have to understand every step of photosynthesis and every step that genomes have to play in terms of photosynthesis. In order to gain a solid understanding of the genome of the target crop and how the specific genes contribute to photosynthesis and growth. we need to understand in depth how all of these different components work together, and also understand how the regulation of photorespiration is happening. Because if we make one misstep, right, one mutation could cause the plant to become how to say, have a lower efficiency, or it could cause a harmful mutation as well. Essentially, genomes are so sensitive that certain regions you can modify entire things, entire sequences, and it'd be fine. But if you modify one base pair wrong in the wrong place, it can make a very bad effect on the plant. So we need to understand where the genomes related to photosynthesis and photorespiration are, how they work, what kind of pathway they take, and how do they all interlink together. Then only we can do the next thing, which is utilizing high level computers with very powerful processing systems to to model the entire thing such that they can find out where they can make improvements. So what are the inefficiencies of the entire system, and where can modifications be made to make it better? So for each crop, there will be a different inefficiency, right? For example, a crop that is close to a very hot environment, the inefficiency is that due to the high temperatures, it is losing out a lot of photosynthesis to photorespiration. So what modification can be made is to modify the Kelvin cycle such that it becomes a C4 or a CAM plant. Okay, so that can be different for different crops, right? Imagine a crop in somewhere like Siberia. That is something else you have to worry about. What are the inefficiencies in photosynthetic properties? The crop that could be found in Siberia is something like frost tolerance. It has not be able to tolerate the frost as well, so that could be an inefficiency. So for each situation, there'll be a different inefficiency that they're targeting. That's what they mean. I'm not changing it. And then the last one is using CRISPR-Cas9 technologies to edit and then, sorry, to target and then edit those genes and create the kind of modifications that we want. So by doing all three of these, we can then generate a genetically modified plant. And it will have the traits that we are expecting. So you know how they say here CRISPR-Cas9 technology is still relatively new? That ain't the case anymore. CRISPR-Cas9 has been around for 10 years now. That is why the first genetic therapy has actually been released. It has always been the case with huge scientific discoveries like this. It takes about 10 years for therapies to come out, for research to actually bear fruit. And we can start to see it's happening, and it's only going to get more and more intense from here. If you need an example of that, think about AI, right? At first, 5, 6 years ago, AI was doing stupid things like making funny videos or making memes, right? Now AI is so powerful enough that it's tricking universities and colleges, and no one can figure out where it's going to end up. That's the same thing with this advancement as well. With CRISPR-Cas9, we have ideas of where it can go, but we have no idea where it's actually going to end up in the future.